University of Liverpool, and the Integrative Genomics of Aging group talked to us today. Worked with the group sequence the Genome and bowhead rather, so talked to us today about the consequences of that in aging.
Thank you. So thank you for the opportunity to tell you a little bit about about what we do. And so, I mean, when I'll do it, I'll cover a couple of different topics in the science of aging, from the genetics of aging in model systems and in humans, also the impact of diet on aging, and also what we can learn from long-lived disease-resistant species to ultimately improve human health. So I guess people have always sought eternal life and everlasting youth. And one of the first documented cases is quite famous. It's called Qin Shi Huang, who was a Chinese emperor. He was the first emperor of the Qing dynasty. He's famous for the ordering the construction of a terracotta army. He's famous for so he's quite famous. As you can see, it was even before Christ, over 2,000 years ago. And he didn't want to grow old. He wanted to live forever. So he asked his physicians, what can I do in order to live forever? And his physicians came up, court physicians came up with this pill and said, take this and you'll live forever. Those pills, the key ingredient that these physicians came up with was mercury. Now, and he did take those pills, and as you imagine, he died of mercury poisoning. Now, things have come a long way since then in our understanding of just toxicity of some compounds, but our understanding of biology and of aging. Having said that, aging is still a mystery of biology in the sense that we still do not understand what are the drivers of aging from a basic molecular perspective. There are a number of theories of aging that you probably are aware of. Oxidative damage, DNA damage. So there are theories of aging, but none of them have been conclusively proven. So we still don't fully understand why we age at the basic molecular cellular level. Which is important because when you think about aging, I'm sure you're aware that there is this graying of the population, and age is a major risk factor for cancer, for heart diseases, for neurodegenerative diseases. So this is a major social and economic challenge on one hand. For instance, it's been estimated by 2050 in Britain there will be 4 times more patients with Alzheimer's disease. So that is an economic social challenge. But on the other hand, it's also a tremendous opportunity, because it means that if we could slow down the process of aging, even if slightly, that this would have unprecedented medical benefits. So now, I just told you that we don't know why we age, however. Having said that, there are some reasons to be optimistic, and I'll give you a couple of different perspectives on this. So the first, which is something that always fascinates me, is that there are species that appear not to age at all. So, as I'm sure you're aware, all human beings age. As far as the whole mammals age, as I'll mention later, there are some species that live longer than us, some species of whales. But there are species that appear not to age at all. So this includes, I'm talking about, as you can see here, I'm talking about complex animals. I'm not talking about amoebas or microbes. I'm talking about complex vertebrates. So there are some species of fish. So this is a particular species from genus Ambastis, which is a type of rockfish. There are some species of turtles. This is, I think, the Galapagos tortoise. This is the painted tortoise. And they appear not to age at all. And what it means by that is that, as far as we can tell, they do not exhibit the typical hallmarks of aging. And these are physiological deterioration. So there's been studies in turtles spanning decades that didn't find any evidence of physiological deterioration. And they do not exhibit that typical exponential increase in mortality that you see in other species. So for example, in human beings, once you reach about the age 30, your chances of dying double roughly every 8 years. And that is a universal phenomenon that happens across countries and across human populations. But again, in studies spanning decades, this has not been observed in some species. And in some cases, not in all, in some cases they do exhibit an increased reproductive output with age. So you see this in some spicy species, for instance. So some species of fish, the animals just go continuously. So the older they are, the bigger they are. So females, for instance, the bigger they are, the older they are, the more eggs they can lay. which you can think from an evolution perspective that maybe makes some sense for these animals not to age at all, because they can reproduce more the older they are. But that doesn't happen actually in all types of the species. The point is that these animals appear not to age. I mean, perhaps they age over 500 years, but at the very least they age much, much slower than human beings. So what that tells us is that aging is not universal. Yes, it affects everyone, but it doesn't affect every species on Earth. And there are complex vertebrate species that avoid it. So natural selection can have a tremendous impact on aging. The other major breakthrough, I guess, in the field in the past 20, 30 years is that the process of aging is surprisingly plastic, both from a genetic and from an environmental perspective, and a dietary perspective. So, I mean, I'll talk about diet in a little bit, but particularly in terms of genes, we now know of dozens of genes that have a significant impact on aging. So, I guess the first gene to be identified to have an impact on aging was called age 1. Well, they call it age 1 because we didn't know what it did at the time. This was Tom Johnson and colleagues at Colorado. And they were working with C. elegans, the roundworm C. elegans. And what they found, it's been found, reproduced by a number of other authors, is that you can manipulate one single gene in C. elegans and it has a dramatic impact on lifespan. So I think initially they found you had about 2-fold increase in lifespan. It's a single gene that you manipulate in worms, and worms live twice as long. And the current record, I think, is about 10 times longer. So again, it's a single gene, and interestingly enough, is another allele from age 1. It's a single gene that if you just have one variant, and worms live 10 times longer, which I guess you could extrapolate to people, that would be people living over 1,000 years. And so, now, my understanding is that there's people in us from different backgrounds. So I thought I'd just include a background, just one quick recap for biologists in the room, but for the others. So, in essence, the proteins are really the machines of life. They're the ones that determine our traits and the phenotypes. The proteins are encoded in the genome, encoded in the DNA, which is in the nucleus of our cells. And so what we try to look for when we talk about genetics is looking at different variants in the genes that in turn determine different variants in the proteins that in turn have different functions in cells and have different actions. And what really is, I think, striking from a genetics of aging perspective is that even though the genome is fairly big, so the human genome is about 3 billion base pairs, there's about 20,000 genes, 20,000 different genes/proteins. You can have very tiny mutations in a single gene that can have a dramatic impact on aging. And that is remarkable because aging is very complex. Aging entails multiple changes and affects virtually every system in the body. And yet, you can have one single change in one single gene that can change the rate of aging fairly dramatically, at least in mortal systems. And so, one of the things that, so when I was a PhD student about, so over 10 years ago, when I was a PhD student, I was very fascinated by this, capacity of genes to regulate aging. You can have one single gene that regulates the rate of aging. And so what I decided to do is, initially I had an Excel spreadsheet of all the genes, and essentially I decided to make a database out of it. It's called the Genome Database of Aging-Related Genes, which now arguably is the benchmark database in the field. And so just to give an idea without going into too many detail. So as I mentioned earlier, in C. elegans, the current record is the greatest lifespan increase is about 10-fold by a single gene. But this is the total number of genes in our database. So as you can see, it's nearly 700 genes. So one single gene can have 10-fold impact, but there are hundreds of genes that have a significant impact on lifespan and on aging in C. elegans. Now, of course, C. elegans is a tiny microscopic worm. My question is, how important it is that for human beings? Well, if you look at mice, which are mammals like us and therefore much more closely related to us, even in mice, there's nearly 100 genes, single genes that have a significant impact on aging. The current record, I think, is for the growth hormone receptor. So if you knock out the growth hormone receptor in mice, it increases lifespan by about 50%, which you're thinking, okay, so that's not as impressive as worms. No, but I think living 50% longer would still be quite impressive if that would be applicable to human beings. And one very important point is that it's not just that these animals are living longer, they're living longer healthier. So their process of aging is retarded in these animals. So they're living longer, and that physiological deterioration is delayed. The onset of age-related diseases is delayed. So the whole process of aging is being retarded in these animals. Which is really what we want, because we don't want to just live longer, we want to live longer with health so that we can enjoy the extra years of life. So I guess the point is that aging is not just a sum of age-related diseases. There are intrinsic mechanisms of aging, because they're still largely mysterious, but it is possible to regulate the process of aging. And when you think about human beings, we cannot mutate genes like we can in mice or in C. elegans, but you can try to manipulate genes by diet, by lifestyle, and perhaps even by drugs. And if we could do this to retard the process of aging, this would have a huge impact in extending lives and preserving health. Now, okay, so we have these genes that we know have an impact on aging or regulate aging in the mortal system. How potentially relevant are they for human beings? As you might imagine, it's not that simple to study aging in human beings. But there is some evidence, at least, that genetics has a significant impact on human aging. And I'll show you a couple of examples. So the first example is actually been fascinating since I was a PhD student as well. It's called Werner syndrome. And as you can see, so this is a patient with Werner syndrome. This is some patient at 40 years of age. So patients in this disease, they appear to be aging faster than normal. So they're older at a younger age than would be expected. And so it's called segmental prodroid syndromes, which are syndromes that resemble accelerated aging. And they have an early onset of a variety of age-related diseases and physiological deterioration. In some systems, it's not perfect accelerated aging, but in some systems resembling accelerated aging. And this disease, it occurs because of mutations in a single gene called the Werner syndrome gene, very creatively. But the point is that you can have mutations in a single gene that have a significant impact on aging. But what about the opposite? Can aging be retarded thanks to mutations? So there's a couple of examples. This is a relatively recent one. It's a quite interesting study. This was done by Walter Longo and Jaime Guevara-Aguirre. So this is Dr. Jaime Guevara-Aguirre, and these are his individuals that he studied. And as you can see, it's not that the doctor is very tall. What happens is that he's studying small people. And the reason they're small is because they have a mutation in the growth hormone receptor gene. Now, the growth hormone receptor gene, as I mentioned earlier, if you knock it out in months, the animals live considerably longer, about 50% longer. And so the hypothesis was, well, these individuals with growth hormone receptor mutations, are they long-lived or are they protected from age-related diseases? So the most interesting result that they found was that these individuals appear to be strongly protected from cancer. So what you see here, so these are growth hormone receptor deficiency. So these are these individuals. These are their relatives that don't have the same mutation. And although the sample size is not that large, these individuals do not appear to die of cancer. Maybe some of them will, but they appear to be strongly protected from cancer. As compared to their relatives in which 20-25% of them die of cancer, which is kind of what you expect. I don't want to be too dramatic, but you expect about 20-25% of people will die of cancer. So at the very least, they are very strongly protected from cancer. Now, our obvious question is, so are these individuals living longer? And the answer is no. They appear to have increase of neurological conditions, certain neurological conditions, and heart disease. So they do not live long, they live about the same as their relatives, but at least at the level of one specific age-related disease, they appear to be strongly protected. Now, the other angle of looking at the genetics of human aging, genetics of humans, is to look at specific longevity. We know that people have different longevity. There are exceptionally long-lived individuals. And so this is your Helena. So she was from the south of France, and she lived to be 122. She's actually a wonderful example of There's a great anecdote about her because she consecutively broke longevity records. She became a celebrity in France. And so every year, she was 110, 111, and so on, journalists would flock to her house and ask her, Ms. Alcantara, what is the secret of your longevity? And she started to be a bit tired of it. And so one year, when she was 115, and the journalist asked her, what is the secret of your longevity? She replied, the secret for me to live to be 115 is that I stopped smoking when I turned 110. And that's a good story because there's actually an element of truth to it. She actually was a smoker for most of her life, and she stopped smoking, I don't know if it was 110, but she stopped fairly late in her life, actually because her eyesight was getting worse, and she was afraid she would hurt herself lighting a cigarette. So it's a good, and she was also fairly fond of port wine and chocolate. So not the type of lifestyle you would associate with the longest lived human being on record. So clearly genetics also has to play a role. And so, one of the things we've also been doing is looking at genes. So you can look at individuals, for example, centenarians, and you can look at octogenarians, and you can see if there's differences in their genetics, in particular genetic variants between them, and try to look for genes that are associated with longevity on being a centenarian. And so we actually also have a database about it, which catalogs all the studies done in this area. What we know about the genetics of longevity is that, so you can look, for instance, studies in twins to determine how heritable a particular phenotype, a given trait is. So what's the impact of genetics in essence? And we know that longevity in humans is moderately heritable. So it's about 24%. But what it means is that how long you live is only about 25% genetics. But there's a caveat, which is it's much higher the older in older individuals. So for example, in centenarians, maybe I'll look 100 or 200 centenarians and try to find do these people eat the same thing. Maybe they eat a lot of yogurt, maybe they go on strolls in the beach. And what they found is that there's nothing in common in terms of lifestyle between them. But many of them have other relatives that are more living in common. So the take-home message is, if you want to live to be a healthy 80-year-old, then you better watch your lifestyle. Don't smoke, exercise, watch what you eat. But if you want to be a super centenarian or a centenarian, live 100 or 110 years of age, really what you need is good genes. So that's what's important to keep in mind. Now the other important thing is that I said earlier that so we know about all of those genes in mortal systems that have significant impact on aging, hundreds of them. We can manipulate aging to very surprising even, and to a very great extent in mortal systems. But when you look about genes associated with longevity across populations, there's very, very little actually that becomes significant. There's one gene that is consistently associated with aging, a gene called ApoE, polyprotein E, which is related to cholesterol, which, as you might expect, is then associated with heart diseases and Alzheimer's disease, etc. But apart from that, we actually know very, very little about why these individuals live longer, why some people become centenarians. There has to be a genetic element, but we still don't know about it. So, for example, one of the possibilities that maybe there are what's called rare variants. So maybe what makes Mademoiselle Joao live to be 120 is different to what makes another person live to be 150. And that is very difficult to identify. But nowadays, you probably are aware, genome sequencing is relatively cheap. So you can sequence your genome nowadays by about $1,000. Well, the original human genome costs about $3 billion, and over 10 years to complete. You can sequence your genome by $1,000 in a couple of weeks. So yeah, the way the field is moving is to really sequencing the genomes of multiple individuals, including these super centenarians, people over 110 years of age, try to identify these variants associated with acceptable longevity. Now, in addition to genetic manipulation, there's also dietary manipulation. So as you're aware, what you eat has an impact on lifestyle. A unless you're really lucky to have whatever genes protect centenarians from unhealthy lifestyles. And we've known that for decades, and we know that diet can have a very strong impact on aging. So the most widely studied in the field and the most consistent intervention to extend lifespan is called caloric restriction. So as the name implies, caloric restriction, it means reducing the amount of calories animals eat without malnutrition. So for those who are not very familiar with animal studies, the way, for example, we keep mice or rats in a lab is something called ad libitum fat. So what it means is that they have food and they can eat how much they want. But what was discovered by McKay and colleagues, following some early findings, is that if you restrict the amount of calories the animals eat while maintaining healthy vitamins and minerals, the animals actually live significantly longer. So this is one study, I think this is from San Antonio, Edward Swarm, I think, studies in rats. So what you see here is a study in rats. So this is the percentage of animals alive, and this is age. Okay, so rats live a little over 3 years. So if you feed them ad libitum, they live about a little over 3 years, as you'd expect. But if you caloric restrict them, they live significantly longer, about 50% longer. And again, just like with the genetic manipulations, it's not just living longer, they're living longer healthy. So it is quite remarkable, the impact that caloric restriction can have on the aging process. So, I mean, this has been known for, as you can see, for decades. And just to summarize what we've learned for the past 50 years of research on caloric restriction, that so we know it extends lifespan in most species, but not all of them. There are a couple of exceptions. It can have negative side effects. So I mean, I don't recommend caloric restriction to human beings. For example, it can be detrimental for fighting infections. So for instance, in laboratory studies, in animal studies, animals are kept in relatively clean or even pathogen-free conditions. Is that you're trying to minimize the amount of infections they get because you don't want animals to die of infections if you want to study aging. But if you actually expose animals under caloric restriction to infections, they will die. They have high mortality. Do you have a question?
Yeah, just wondering, in your example of the calorically restricted rats, they did die more frequently in the first 2 years. Is that because of?
There is a slight, you mean here?
Yes.
I don't know how much of an impact it is that, but it has been observed actually that I have seen it in our study. I think it could be actually infectious diseases. I don't know for sure what the animals are dying of, but I have seen that kind of effect. When you put animals in caloric restriction, it increases slightly mortality at the beginning before the levels of non-mortality is reduced. The way you would think of caloric restriction actually is it's a stress. It is stressful for the animals. It is stressful for people as well. There are people who try to undergo caloric restriction. So it is a form of stress. And even young animals, if you stress them, some of them it could be fatal. So that's the way I interpret it. But you're right to point it out. It can, at the beginning, it can be detrimental.
Do they produce as many offspring? I mean, they may not be as fit classically as
That is a good question. Yes and no. Okay, so noted on producing enough offspring. So if you take age-matched, relatively young animals, say here, if you look at these animals, caloric restriction and ad libitum, the ad libitum are going to be more fertile. They're also going to be bigger and more fertile. However, the reproductive age of the animals, the reproductive lifespan of the animals is increased under caloric restriction. So, in other words, older animals under caloric restriction can reproduce, while animals fed ad libitum can no longer. So, there's a bit of a trade-off. On one hand, their reproductive output is decreased initially, but they can reproduce for longer.
But maybe in the wild they don't do as well because they're competing with bigger rats or whatever.
I don't think caloric restriction would work that well in the wild unless they were in very specific environments. I mean, there are cases, not in rats as far as I remember, right? Cases of fish that people think, well, maybe these animals are under caloric restriction naturally in the wild, in very specific ecological niches. But caloric restriction, the way here, I would see it more as an artificial process. I wouldn't see it as something that normally occurs in the wild.
I'm just trying to explain in my mind rats have been optimized for the alleles of genes that they've got? Why haven't they optimized themselves over evolutionary time to live longer and be healthier? There must be some reason for it. If they're not as fit, then
Yeah, that's a good point. I think there's a couple of reasons. The first one is that rats and mice in the wild typically do not live more than 1 year. So, mice and rats would live about this. This is where they would die. So, living for a rat or a mouse in the wild to live 3 years or 4 years doesn't make a difference because most of them are going to die long before that. And there are trade-offs in caloric restriction. As I mentioned, the animals are smaller. Particularly male mice, for example, and rats, you tend to fight a lot. So if you're going to be weaker, you're going to be at a disadvantage considered against the others. So there are trade-offs in caloric restriction. Another negative side effect would be that it is detrimental for fighting infections. We don't fully understand the mechanisms. There's hormone alterations that are important, but molecular mechanisms are not fully understood. And there's a very long-term study going on caloric restriction in rhesus monkeys, which you see here. Now, rhesus monkeys live over 40 years. It's been a very long, very expensive study in the US. And it's actually having conflicting results because there's actually 2 labs doing it. And both of them suggest that caloric restriction has some health benefits. One of the studies suggests that it will increase lifespan, even though the study hasn't finished yet. Well, the other study suggests it will not increase lifespan. So neither of the studies has finished so far. So the potential benefits to humans are still not very clear. There are, in addition to diet, there are also, in addition to caloric restriction, there are other types of dietary or nutraceuticals that can have an impact on aging. This is probably the one thing gather most attention by researchers in the field, which is rapamycin. So rapamycin is an immune suppressant. It's also used as anti-cancer treatment for particular types of cancer. It needs cell proliferation. And what was found by a large-scale study in the US was that if you feed mice rapamycin, it has a significant impact on lifespan. So what you see here, these are females, and these are males. I think it's quite interesting that these are animals that were nearly 2 years of age when they started feeding them rapamycin. So these are the animals that have rapamycin, these are the controls. So it has a significant impact. And now there's quite a bit of research done on rapamycin, seeing potential applications in humans. Now, my own view on, particularly about caloric restriction, is that it's not going to have the same benefits in humans as it has in rodents, partly because of the studies in monkeys, partly because we humans are much more diverse, heterogeneous group than mouse or rat strains. But it can help identify drug targets. So there's rapamycin, which targets stores. So this is sort of my own view of how caloric restriction works, which is via the effect on the brain that has hormonal effects, and then there's a signaling cascade. Too many players for me to mention here. But even if it's not going to be targeted in humans, it can be used for drug discovery. There's the example of rapamycin. And some of you might have heard of resveratrol, which was found to extend lifespan in mice, although it was later found out it was resulting in obese mice. And resveratrol is a compound that's found in red wine, so sort of that how to explain, for instance, why French people live longer because of high red wine consumption. Well, I would say that the amount of resveratrol that they use in order to get that amount from red wine, you have to drink about 150 bottles of red wine a day. I know I can live a little bit more. Resveratrol only extends lifespan in very specific strains, namely obese strains. But we're interested in essentially trying to look at the genetics of caloric restriction and which genes and which molecules could mediate its life-extending actions. So one of the things we made was a database of genes associated with caloric restriction. You probably noticed a trend that we make a lot of databases. I should explain. So one of the reasons we make a lot of databases is because, well, actually, I think everyone should be studying aging. Every researcher in the world. And even if that's not going to be the case, because I appreciate there's other interesting topics out there. If people are interested in studying aging, I want to make it as easy as possible for them to go into aging and find information and data to do studies of aging. So we made this database of genes related to caloric restriction. So there's particular mutants that don't respond to caloric restriction as well. In terms of lifespan effects. And then one of the things, without going into great details, that again, there's 150 genes associated with the effects of caloric restriction. But just like there's hundreds of genes associated with aging. And one of the things that we're trying to understand is, okay, so there's 20,000 genes in the genome. There's hundreds of genes associated with aging. There's dozens of genes associated with caloric restriction, but how can we understand how they interact with each other really to determine aging or longevity or even other diseases? So I think that's one of the big challenges now in the closed genome areas, is trying to understand how genes interact with each other and really how they determine phenotypes based on their interaction with each other and with the environment. So you can make things like networks. So again, without going into great methodological detail, each of these is a protein. And you can try to look at how they interact. And you can analyze these interactions. And you can link it to other kinds of data like gene expression data, like to try to understand. And just to give one example of one thing you can do something called the guilt by association method, which is something very simple. It can be applied to those diseases. It can be applied to society. So in essence, if you have a number of, let's say, these are proteins that are associated with caloric restriction, and these are their interactions. If you have proteins that have not been associated with caloric restriction, but interact with proteins in turn associated with caloric restriction, you would think that these ones would be good candidates for also being associated with caloric restriction, guilt by association process, which can be applied to other, even non-biological processes. And so we did this, for instance, and we showed that this in true. We tested 9 candidate genes in yeast, and we showed that 8 of them were related to lifespan effects of caloric restriction. So, I mean, the message is that, the genome is complex, biology is complex, and we're starting also to have the computational tools and the methods to decipher that complexity and start to understand how the genes link to each other. And in this case, identify candidates. And just kind of as a side note before I move on, one of the things we're also interested in is trying to identify drugs that affect longevity via caloric restriction. So the same way we can link genes to genes, there's a lot of data sets that allow us to link genes or proteins to drugs. So we essentially try to look for drugs that have a similar effect on the genome, the gene expression level, than caloric restriction. And when we tried it, the top drug was rapamycin, so we tried 1,000 drugs, and the top drug was rapamycin, which, as I mentioned earlier, extends lifespan in mice and in worms and in flies, for instance. So we may say, all right, so maybe there is something to this. Maybe it's not just random, but we are finding something that is related. And so we've got a number of candidates which we've now been testing in C. elegans to see if these drugs extend lifespan in C. elegans. And if they extend lifespan in a worm model of caloric restriction, there's something called yeast mutants. So these are worms, basically they have a defect in their mutant, they have a defect in their larynx, and they physically cannot eat as much. So they're essentially under caloric restriction because of this genetic mutation. So we feed them the drugs to see if they live longer or not, compared to the controls, trying to identify new drugs that extend lifespan. Now, so I talked quite a bit about the genetics and about variations in lifespan and how we can manipulate lifespan within a species. But the other topic that I've always found fascinating is the diversity in the pace of aging across species. So I mentioned earlier that rats live about 3 years, so mice and rats live about 3 or 4 years. There's some exceptional cases and caloric restriction with mutants that can live longer, but about 3 or 4 years. And they are old when they're 3-year-olds. So it's not just they're dropping dead. They are, if you look at them, they look older and quite aged when they're 3 years of age. Monkeys, which I mentioned earlier, can live 40 years. Humans 122. And at the other hand of the spectrum, you have whales, which have been estimated to live over 200 years. So there's this huge variation in longevity amongst mammals. even among primates, there's very strong variation. Not just in longevity, but in rates of aging. So rhesus monkeys will age significantly faster than humans. Chimpanzees, even though they're our closest relative, they will age significantly faster than human beings. And it's not due to metabolic rates. I mean, there's this notion that it's called rate of living theory, that animals that have a higher metabolic rate are going to accumulate damage faster and are going to age faster. And this was, I mean, actually dates back to the Industrial Revolution, where people noticed that machines, if you run them faster, they're going to last, not last as long. And they thought it could be applicable to animals as well. I mean, we and others have shown that that's not true. I mean, there's a lot of species like bats that have a very high metabolic rate, and yet live very long. So bats can live over 40 years. So that's not true. I'm not going to publish that. Other people actually lost, have done, John Speakman have done the same. So it's not due to metabolic rate. So there has to be other physiological, molecular, and importantly, genetic differences between organisms to determine the rate of aging. So we've been doing a couple of works on this. It might not surprise you that we also made a database. So this was one of my earlier databases called NH, which is a database of essentially longevity records in animals. So I was always been fascinated by this diversity of longevity across species, and so I started recording longevity records. And again, I thought, well, why not just put them online on a database instead of just having this Excel spreadsheet on my laptop? So we have over 4,000 species and over 1,000 mammals. So it's a database of longevity records. It also has metabolic rates and life history traits, things like body size. And then, so this allows us to do a number of comparative studies of aging. And then we've also been focusing on a couple of species of interest. The first of them is Nick and Mulder. And I mean, we're not just interested in them because they're beautiful. We're interested in them because they're the longest lived rodents. So they can live over 30 years. So these are, they're a little bigger than a mouse and smaller than a rat. So they're relatively small animals. They live on the ground. So they can live over 30 years, which is quite remarkable when you compare it to mice and rats, like 3 or 4 years. But I think what's even more remarkable about them is that they're extremely cancer resistant. So, over 1,000 animals kept in captivity. So far, there's not been a single cancer case. Maybe there will be one day, but at the very least, they're extremely resistant to cancer. So back when I was a postdoc in Boston, we put in a genome sequencing proposal. So just to put in perspective, I think we asked for, we were saying $15 million to sequence the genome in 2007. And they said it was too expensive. But since then, a couple of different labs have sequenced the genome. So the BGI sequenced it first. And together with Broad Institute, Harvard and MIT, we sequenced it as well. So, I mean, again, we made a database out of it. So, if you're interested in the genes of long-lived species, this is online as well. And then we're trying to find particular genes that may have evolved different functions and may explain the cancer protection of the nickel. One of the genes we focused on was p53, which maybe where it's very commonly mutated in cancer. It's one of the most widely studied genes, actually. It's probably the most widely studied gene, at least in the context of cancer. So we found particular changes in p53 that could be related to cancer protection of the nickel. But again, all of the data is online. My objective is that more people, there's more research done on the short-lived animals, but I like to think that the long-lived animals will also have important insights about aging, longevity, and disease resistance. And there's a lot of untapped potential in these long-lived species. So the nickel would be an example of this long-lived, disease-resistant animal. In addition to that, and more recently, we also focused on the bowhead whale. And so we became interested in the bowhead whale again because it lives very long. So they've been estimated to live over 200 years. And it's also interesting because they're the heaviest animal, or one of the heaviest animals on Earth. So they must have some sort of anti-tumor mechanism. So if you think about cancer as emerging from one rogue cell that starts proliferating uncontrollably, then an animal like this one that has 1,000 times more cells than human beings must have 1,000 times higher chance of developing cancer. And thus, you cannot live very long. But of course, they do live very long. And so they must have some sort of anti-tumor mechanisms. Now, Well, there's a couple of reasons. I mean, we don't really go to birthday parties. But we do know that, so for instance, there's some anecdotal evidence that, for instance, there's been whaling artifacts. I mean, this is the tip of, what do you call it? Those things, hunt whales.
Harpoons?
Sorry?
Harpoons?
Yes, it's the tip of an harpoon that was found in a whale. Whales are still, so bowhead whales, there's an annual hunt in Alaska, and also occasionally in Greenland, in Russia as well, and in Canada, by native peoples. And so occasionally they found these whaling artifacts that have been estimated to be over 100 years old. So that kind of suggests that these animals that were killed down were over 100 years out there. And there's also a biochemical method to estimate how long these whale are. I mean, without going into great detail, basically this is based on something called a decimation ratio in the eyelids. So you look at the eyelids of the whales, it's like a tree trunk. Look at these older ones, and you can look at these biochemical properties of their amino acids, and you can estimate based on known ages of whales, you can estimate how old the oldest individuals you find are. It's an indirect method. So you're basically estimating how old this individual is. So one individual is estimated to be over 200 years of age. That's a big error margin there. Even so, I mean, I think when you take everything together, I think it's reasonable to think that these animals can live longer than human beings, and in the wild, without medical care. So I think that's very impressive, that they must have some sort of anti-aging and anti-cancer mechanisms. So we sequence the genome again without going into great details. So what you do when you sequence the genome, when you sequence your genome, if you're interested, is you take DNA out and then you chop it into billions of little pieces. And you sequence all of those billions of tiny pieces, and then you put it all together in a big computer as a big, massive jigsaw puzzle. And then you find genes, and we find like 23,000 protein coding genes in the genome. But then really what we want to find is genes that could be related to the exceptional traits of the bowhead whale. Then we found a few candidates. I'm not going to mention them all or go into great detail. So one of the things about the bowhead whale is that its genome is actually fairly similar to that of other whales. And most of all, so we compare, this is the bowhead whale, we mostly compared it to the minke whale, which is a much smaller and presumably shorter-lived whale, and also to other cetaceans like the killer whale, which is not a whale, it's a dolphin, and the dolphin. And so we're then trying to find, in the bowhead whale, we're trying to find genes that have differences that no other species closely related to it has. That's what we're trying to do. But the genomes are actually fairly similar. So the bowhead whale genome is actually fairly similar to the minke whale genome. But we managed to find a few genes, in particular genes that have known to be associated with cancer and aging. So to give one example, this is pCNA, which stands for proliferated cell nuclear antigen, which has been associated with cell proliferation, as the name implies, and also with cancer and with aging. And we can find a few very specific mutations in pCNA of the bowhead that with three-dimensional structures, we can predict they're going to have a phenotype. Of course, we cannot go back to whales and study them. I don't think my head of the department is like that. But we can try to look at, what we're trying to do now is we're trying to look at these genes in cell lines or maybe even in mice. So for instance, you can take a mouse and you can make a mouse with a bowhead whale gene or with these specific differences here and see if that has an impact on the longevity and cancer resistance of the mouse. So that's what we're trying to get funding to do now. And so we found several cancer and aging related genes. We found many other genes. Papers published in January. We found quite a lot of different changes. One more that I thought was quite interesting. So the whales, they're massive, and it's been estimated that if the cell metabolic rate, essentially, the energy released by the cells of the bowhead whale were similar to a human being, they would literally boil the water around them because they would generate so much energy. So they must have some sort of different thermoregulation when compared to human beings. One of the things that was interesting was that we found a massive deletion in the major gene involved in thermoregulation in animals, which is UCD1. So this is the sequence of the protein in cow, human, and mouse. And these are 4 different species of whales, and they have the sequence is like. And these are actually the active, or 2 of the active sites of the protein in mammals, as far as we know, and whales lack. So again, we cannot really create a mutant whale to test this, but it does seem to indicate that whales have differences in thermoregulation. They could be related to this gene. Now, I want to mention, so we published this and quite a bit of media coverage. And there's a couple of, I think, interesting lessons on that as well. So it was covered in newspapers in Britain and Spain, Austria, Germany, Portugal, etc. One of my students' favorites was the I fucking love science website, which you might be familiar with, which got 35,000 likes on Facebook, which my students enjoyed. Although I have to say that there was another story that same week that got more likes, which was the man with two functional teams. Now there was one of the things I enjoy is to go through media coverage and look at the comments from the people on the news featuring our work, because I think it's quite intriguing in some cases, but it's also important to understand the public's perception of our work. So you have the usual jokes like they'll be so long because they don't get married. You have the jokes you might expect. But you also have comments which I think reflect reasonable concerns of the public regarding research on aging. People who would want to live that long. That is a notion, and I think there's one that the researchers on aging have to counterbalance. So when we have to make it clear, I think one of the big concerns of these comments is, you're living longer. And if we extend lifespan, you're going to extend lifespan in the period of decrepitude or dependence. And that's not what aging research is about, and that's not what the animal studies show us, for instance. As I mentioned earlier, you can regulate aging in mice in a way that retards the whole aging process, retards disease and physiological deterioration. But that's still a major misunderstanding, I think, about what we do. That I think we have to address. And then there's the comments people talking about. I like the response, which is, you think we're trying to torture whales for them to do the sexual longevity. There's still some sane people out there. So we published, and you might be surprised to learn that we made a database with the bowhead whale genome. So if you're interested in any gene, and so the genes are also mapped to their human homologs, to their human equivalents. So if you're interested in any gene in context of longevity, context of any trait that could be related to the bowhead whale, all of the data is available online. If there's any issue, please drop me an email. But I would emphasize again that the really goal is that this is a new paradigm in biomedical research, in the sense that we're looking at disease-resistant organisms to identify genes, processes, and mechanisms that protect against disease. It's not what you usually do in traditional model systems. In general, we try to find mechanisms that cause disease. Here we're trying to find mechanisms and genes that protect against disease. And ultimately, we can use this information to intervene in the process of aging, to extend lifespan and preserve life. Again, to extend healthy lifespan, not to just extend lifespan, but to retard aging and make people healthier for longer. That is the goal. Again, I think the message we need to repeat over and over. And so that's that. So, in summary, I told you that aging remains a mystery of biology, which has a major impact on society and medicine. But if we could manipulate it, we would have an unprecedented impact on medicine, on society, and the economic impact. I told you that individual genes can regulate aging in mortal systems, and there's hundreds of them that we know now. But it is still a challenge, I think, the level of human longevity, by contrast, because understanding the genetics of human longevity is still fairly poor. And I also told you that even though there's hundreds of genes associated with aging, I think think one of the big challenges now is to understand how among these hundreds of genes, in context of the 20,000 genes in the genome, how do they interact and how do they, what's their hierarchical regulation, which genes regulate which. And I showed some examples for caloric restriction. And last time I told you about the molding species to identify genes and mechanisms that protect against disease. So thank you very much. I'll just, these are the people who did the work. I guess Michael did most of the work on the bowhead and on the bowhead. Together we had lots of collaborators from Alaska to Greenland to get samples to US, Spain, and Ireland. Robbie and Jingwei and Thomas here, they did most of the work, the recent work we've done on our databases. And so essentially these were the people who did the work that I've told you about. These were the institutions who paid for the work that I've told you about. So I'm fairly easy to find online, but in case anyone wishes to drop me a line, please feel free to do so. Thank you very much for your time and attention.
You mentioned that certain organisms don't seem to age like humans do. What do their mortality curves look like? Do they look like a reverse S? So you have this drop down and then just a plateau that the old ones just keep going on and on in a lab?
Well, there's a couple of different examples. Normally you have a big spike in early life, right? Infancy, birth, and then it drops. So some of them it just remains stable instead of increasing like it happened in humans and mammals. It just remains stable. And some species, it actually continues to decline as far as we can tell. The mortality rate actually declines with age. So there are examples of species like that, but they live in very specific niches. But one caveat, of course, that a lot of the studies are species live decades at least, maybe hundreds of years. So our ability to do studies in that is quite limited, as you might imagine.
So they only appear to not age?
Well, they appear not to age as far as we can tell. They don't age as far as we can tell, but the obvious criticism is, okay, you haven't studied these animals for 500 years.
Yeah.
It's hard enough to get grants for 3 years.
Yeah, so we know that if you keep cultured cells in higher oxygen concentration, they'll senesce more rapidly. And we also know that people who live at high altitude with lower oxygen concentration in the air, those people tend to be generally healthier and possibly have lower incidence of cancer. So I wanted to ask, is there some sort of a link between too much and not enough oxygen and lifespan?
I think in human studies that's okay. So there's a couple of different ways of looking at it. I guess in cells, you're right. Yes, I've actually done that in my PhD ages ago. That if you culture cells with lower oxygen, they'll have a longer proliferative lifespan than higher oxygen. That is true. In human studies, I have seen some works. I haven't seen anything that was very completely conclusive to me, because there's a lot of confounding factors. Maybe people that live at higher altitudes have different lifestyles as well, independently of the oxygen concentration. So I don't think it's that conclusive. And even in animal studies, there are studies that show that. I think this is in frogs. There's a study in frogs, I think in Switzerland, that show that animals at higher altitudes live longer. But animals at higher altitudes also have a lower temperature, and that also correlates with longevity. So, or there's a correlation. Animals at higher altitude maybe have fewer predators. So it's really, I think, difficult to say. As far as I know, I'm trying to think, has anyone has kept mice like hypoxia? Well, I guess hypoxia would kill them, but that wouldn't really be aging. Right? I guess naked morons, you could, some people have argued that naked morons, they live longer, or because of the low oxygen. That doesn't appear to be true, because they live longer, well, they still live very long if you keep them at normal oxygen. Having said that, there could have been adaptations to the hypoxia environment that contributed to their cancer resistance. That is a possibility.
It's kind of a little bit related to that, and the oxygen use and the relationship with that and metabolic rate. I was just wondering if you could say a little bit more about that study, because I was kind of surprised that that there wasn't a relationship, because you gave the example about the whales having a lower metabolism necessarily because of their size, and it seems as though the smaller animals have higher metabolism, but they also have a lower lifespan. So I would think that there would be a link, but apparently not.
Well, okay, so to develop that a little bit, so first of all, one piece of background in a second. So Nick and Morans have a low body temperature and a low metabolic rate. So that would fit into that paradigm. I guess initially people compared rodents to humans, and rodents do have a higher metabolic rate than humans. The issue, there's a couple of issues then, is that first of all, there are long-lived animals, certain species of birds, bats, that have very high metabolic rates. So that is the first question. The second question, more what we did is, so how do you determine metabolic rate? There's a couple of different ways. The most widely used way is oxygen consumption or rest. So you take an animal, you look at how much oxygen it's consuming, which is of course directly proportional to the amount of slides. An elephant is going to bring in and out more oxygen than a mouse, just because it's bigger. So you have to correct for body size. Then you run into a problem, which is body size in turn correlates strongly with longevity. There are exceptions, but bigger animals like whales and elephants tend to live longer than voles and mice and rats, as you would expect. So there are issues in how you do this control, because if you simply divide oxygen consumption by body size, then you end up having a correlation between metabolic rate and body size, which in turn correlates with longevity. So what we did in that study was using modern statistical methods, essentially, probably controlling for body size and controlling for phylogenetic independent contrasts, to look at the correlation. And what we found was that there's no correlation at all between metabolic rates and longevity in mammals or birds. There's a more recent study showing that that is true except for rodents. Within rodents, there is still a correlation between metabolic rate and longevity. It was done by another lab, but I think either way, generally speaking, that correlation does not appear to hold. Does this answer your question? Or I would suggest have a look at the paper. Maybe we could continue the discussion.
Just another question. Regarding you mentioned the multiple genes involved in longevity. I think it's about 90 plus for mammals, giving a 50% increase. Does it multiply if you have all these changes? Can you get sort of 50% times 90? And
that's a good question. So, I mean, in mice, that's, as you mentioned, a very difficult experiment to do, because mice live 3 or 4 years. So, that kind of experiment will take many years.
you get two of them quite easily. Just bring them together.
You can, just breeding itself, that takes time. So, and there are problems. So, the major pathway associated with aging in mice is the growth hormone pathway, like growth hormone receptor. And when you disrupt that pathway, you have small animals. Small animals do not breed as well as big animals. Some of it is just physical. So it's not that simple. There are examples, but there are examples from mutations in worms where people have done that. And the answer is yes, it can. It is synergistic in some cases, and so it is cumulative. You do two mutations, you put them together, they live the same as the sum of the two individuals together, the two mutations together. But you also find mutations that are not cumulative, which you assume to be working in the same pathway, for instance. So the answer is yes, particularly in worms. There's been experiments showing that some of the mutations can have a cumulative effect. A couple of them which
So supposing there was genes found in humans in these supercentenarians that is likely to be connected with them living so long. I believe people have been looking for them and haven't reliably found anything yet. But supposing
individuals that have an extremely low cholesterol levels because of mutation in this very specific gene. And so all the big drug companies are trying to identify drugs that inhibit that particular protein in order to lower cholesterol, because that's a big market. So I think drugs would be normally the way to go, but it depends. There's proteins that are druggable, there are proteins that are not druggable. So I think it would depend a little bit on which gene you end up with. I guess the first thing is to find those genes. I mean, as you mentioned, that's been quite difficult. There's one study published where they sequenced 17 supercentenarians, that are people over 110 years of age. They didn't find any mutations in common between all of them. There's more to improve. So we're doing actually some work with a company called we're doing some work with which is a company that's focusing exactly on genomics of supercentenarians. Do I make that one? Non-disclosure agreements. I can't go into great details on that.
So, doing that kind of network analysis, how do you handle noise? Because that's something that's been kind of cropping up in a lot of domains. That actually our scientific databases, the data there's a surprising amount of noise, both in terms of actual noise in experiments, but also reporting noise, biases, and misreporting. So when trying to construct these networks, one has to actually take into account that these links are very probabilistic.
That's absolutely true. It's a problem we face all the time. I mean, there's noise and there's biases by researchers. I think in networks, so there are sources of protein-protein interactions that can be unbiased. Genome-wide screens that you can use. But there are other types of data which are just based on scientists studying their favorite protein. So B53 has thousands, I think, of protein interactions. Partly because it does interact with a lot of other proteins, partly because it's a very well-studied protein. So that is something, so we actually, one of our students, Maria, she's actually been doing some network analysis. One of the things she takes into account is the number of publications for each gene in a network. Because we assume that genes that are more studied, of course, they're going to have more interactions and they're going to have more associations. So that's one of the things we're trying to take into account. There's really no right way of doing it, just something you have to keep in mind.
Hi, thank you for the interesting talk. I'm particularly interested in resveratrol. And apart from drinking copious amounts of wine, one could take it supplementing. I just wondered if you had any views on that.
So resveratrol, I briefly mentioned, so it does extend lifespan in specific strains of mice, but not in normal mice. So there was a very big study by the same guys that studied rapamycin, which is it's a massive study with hundreds of animals. We're doing a project with them as well, on fish oil, by the way. So they did this study on normal mice, and they found that resveratrol does not increase lifespan in normal mice. So my own view is that it doesn't. I would not recommend. I would not recommend taking resveratrol supplements. I'm trying to think, does it have any side effects? You get to have very expensive tea. Other than that, I don't think it has any effects either. So I'm relatively skeptic about resveratrol, I have to say. Go ahead.
So I think that the aging is the big risk factor for neurodegeneration. So that's why the human is probably the only species that develop Parkinson's or Alzheimer's. So like in the newer animals that live like a lot longer than they're supposed to be, like the rats that live like 3 years or monkeys like living like 40 years, do they develop any kind of like similar symptoms to Parkinson's or neurological disorders?
So as far as I know, in small rodents only develop neurodegenerative diseases if they're mutated to develop neurodegenerative diseases. So there's Alzheimer's models, but they have upween tau, the other one, I forget, mutations. So normally in rodents, no, the answer is no. Neurodegenerative diseases in primates, it's a very controversial topic. There's been very few studies, very expensive to study as well, because you need old animals, and it's not that simple thing to study. So I have seen papers, trying to think of I can't remember the papers in rhesus monkeys, but I have seen papers on chimpanzees and on marmosets to suggest, similar, Alzheimer-like tangles in the brain. But I haven't seen papers on cognitive function. So I guess that's probably also difficult. But so I think it's a controversial one. My own view is that they might develop something relatively resembling neurodegenerative diseases, but probably not exactly the same.
Has anybody looked at their brain, for example? Like, is there any loss of neurons or like reduction in their brain sizes or something like
yes,
that?
I think so. In chimpanzees, you do see that. Whether it's Alzheimer's or not, it's kind of a different question because you can see those in normal aging as well in humans without Alzheimer's disease. So that's the big question mark, and I don't think it's known. Okay.
How does immune suppression sort of slow down aging? Is that to do with decreasing things like inflammation in old age, or is it something else?
Immunosuppression. How the immune system changes with age.
Well, you mentioned that rapamycin, for example, is an immune suppressant.
Yes.
So how does that actually prevent aging?
Well, rapamycin is an immunosuppressant. So there's two issues. So rapamycin does a lot of things. Some of them are good, others are bad. So it does have immunosuppressant properties, and it's used for organ transplantees, for instance. That doesn't seem to be related to its life-extending properties. If anything, it's a side effect. And actually, what labs and companies are trying to do is to find drugs that do the same thing in rapamycin without the immunosuppressive effects. I mean, there's some work done on the TOR complex because there's two different pathways, TOR1 and TOR2, which seem to do two different things. There is sabatini now, for instance. Sorry, they might use it. So that's the immunosuppressant. But then you have a lot of immune system related changes as well. Increased inflammation, which is one of the hallmarks of aging. It's observed in, as far as we know, all mammals. And then so you have inflammation and a decreased immune responses to infectious diseases, which is why, as you probably are aware, the common flu is actually one of the major killers in older individuals because the immune system is worse. So you have those two angles. Now, there was a kind of linking the two of them. There was a recent work by Novartis, their branch in Cambridge, not Cambridge here, sorry, Cambridge, Massachusetts, where they gave rapamycin to these elderly individuals, and they showed that their immune responses for a brief period of time, and after that they showed that their immune system was functioning better, so it improved functionality, which is kind of a proof of concept that rapamycin may have clinical applications in the elderly. But it was a relatively small study, relatively small number of individuals. I think it was at New Zealand actually, individuals. So those are different angles. So exactly how rapamycin can be used, I think it's still a big question mark.
You mentioned some of the animal species that appear that are not age, we have identified with aging age. Is there any insight or what insights are there into how they go about not aging?
Not really. I mean, most of them are very difficult to study. I guess there are some. I mean, like very simple animals, like Hydra. So Hydra is this really tiny thing. So it's an animal-ish. It just gobbles up, I think a little plant, microorganisms, and it's basically made up of stem cells. So it can regenerate. It says the external regenerative potential. It can regenerate the whole body and so on. And there's other animals like some species of jellyfish, etc. These are very, very simple. So those ones we kind of understand. They're so simple they can regenerate the whole body. But when you're talking about examples I mentioned like turtles, I mean the answer is no. We don't really understand. We can look at some aspects. So for instance, I think some of them have very they seem to be very stress resistant to particular types of stress.